Spooner, and I'm the chair of the Patient-Centric Sampling Interest Group. I'd like to welcome you to our latest webinar featuring Chiara Cancellerini, Cancellerini from the University of Bologna in Italy. So sorry, Chiara. Entitled Remote Monitoring of Antiseizure Medications at Home Through Microsampling Devices in Epilepsy. Just before Chiara starts to talk to us, just like to thank our partners and sponsors who fund the Patient-Centric Sampling Interest Group and enable us to have our webinars every month. All of our webinars are available through our website and through our YouTube channel. We will hopefully have time for your questions to Chiara. Please can you submit those through the chat function on the platform we're on, and note that the webinar is being recorded and will be available in a few days through our website and YouTube channel. And with that, Chiara, I'll hand over to you, and you'll need to take control of the slides. Thank you all.
Thank you, Neil. Thank you. Good afternoon. Thank you for this amazing opportunity to share our results. Today I will present the remote microsampling monitoring for antiseizure medication at home through capillary microsampling in adult patients with epilepsy. So my presentation will be structured into four main sections. First, I will cover some background in epilepsy, then the pharmacology of antiseizure medication, and then I will discuss the role of therapeutic drug monitoring, or TDM. And then I will move to our primary research project and the development of population pharmacokinetics model. Finally, I will briefly conclude presenting the next step in the future direction in research. So first of all, epilepsy is one of the most prevalent chronic neurological disorders worldwide that affect 50 million patients across all age groups. And clinically, it is characterized by a recurrent unprovoked seizure that is caused by a sudden and abnormal electrical activity in the brain. Patients frequently experience also psychiatric comorbidities such as anxiety or depression, but also social stigma, discrimination that can restrict their access to education and employment. And actually, unfortunately, epilepsy presents a profound global socioeconomic disparity because nearly 80% of the individuals live in low and middle income countries. So as you can see in this schematic of the GABAergic synapses, when inhibitor tone fail, it triggers a sudden and extensive electrical discharge. And this is when a seizure results as a sudden and extensive electrical discharge within a group of neurons. And this is the seizure generation that is the core of the phenomena, where is the neurological hyperexcitability, which a disruption of the delicate balance between the excitation and inhibition. And because the underlying pathophysiological mechanisms are different, we can require distinct pharmacological target. We can either enhance GABAergic inhibition, attenuate the glutamatergic excitation, or modulate the voltage ion channel. So when we're looking at the treatment of the epilepsy, we have an incredible therapeutic evolution over time. Phenobarbital was introduced in the early 90s as a barbituric, and it works primarily by enhancing GABAergic neurotransmission. But a major turning point occurred during 60s and 80s, which saw a real boom in first generation drug. And this brings all the second generation that was born the French valproic acid, carbamazepine, but also phenytoin. And this medication provide actually clinicians to have a real different mechanism of action And then the third generation of agent over the 2 decades was highly effective to better tolerate drug like lamotrigine, levetiracetam, topiramate. And more recently, the therapeutic landscape has been further enriched with the cenobamate, which highly efficacy in focal seizure, fenfluramine, and fenfluramine, that is a fascinating case of a drug that was originally developed for appetite suppressant and now is reproposed for a rare syndrome of epilepsy, like Dravet syndrome and Lafora disease. So it's true that we have a broader spectrum of antiseizure medication, but actually modern therapeutical strategies can successfully achieve a complete seizure control up to 60% of patients. But approximately 30% of patients suffer from refractory epilepsy. So pharmacoresistance remain a great clinical challenge. Indeed, initial monotherapy bring a seizure freedom in the 50% of the cases, but when starting the add-on therapy, the probability to achieve seizure freedom decrease significantly with every failed treatment. And this is why clinicians can consider alternative strategies such as surgery or different type of diet. So these, because we deal with such a high percentile of complex polytherapy patient, optimize treatment on a highly personalized doses become really crucial. And this is bring us to the second part of the presentation of today's agenda, that is the therapeutic drug monitoring, or TDM. So what is TDM? TDM involves the measuring of the concentration of drug patients in the biological samples to ensure that they remain within a specific predefined therapeutic window. And this approach is vital for 2 main reasons. So to prevent the dose-dependent drug toxicity on one hand, and also to ensure that the clinical efficacy is maintaining. And of course, TDM is needed to guide the clinician to adjust the dosage, the drug dosage, based on the individual patient's need. So why TDM in epilepsy? Beside the routine TDM, it becomes really important after a breakthrough seizure that can allow us to determine if the event was triggered by as underdosing of or a poor patient's compliance, so we can better understand what happened. And is really important also when patient experience adverse effect, such as a classical sign of lamotrigine that I reported here, or to monitoring also a drug-drug interaction with we can be really frequent, for instance, in women taking extragenal oral contraceptive. So for go deeply, how TDM is done. So for do TDM, we use a biological sampling. The blood collection by venipuncture remain the standard approach to measure total drug concentration, but also alternative matrices are becoming interestingly important, like saliva, urine, or breast milk. Once collect the biological samples, the laboratory quantification require extreme precision. And today, the gold standard is the high-performance liquid chromatography mass spectrometry, so HPLC-MS, because this technique can offer high sensitivity and simultaneous multi-drug quantification. And then the pharmacist and clinician consultation access the patient personalized dosing and see if the dose is well described. So which are also the challenge in TDM, because of course we have some clinical and logistical challenge. And first, there is an issue of the invasiveness. This because the traditional venipuncture can be painful and induce anxiety or nidophobia. And this is the major problem when treating patients, pediatric patients, but also uncooperative patients, such as encephalopathies, or with the rare disease, as I showed you before. And moreover, patients must travel to go to the specialized hospital, and these incur time and financial costs for themselves, but also for caregiver, which can create a huge disparity for those living in the remote area. For instance, in Italy, the third care hospitals are located in the main city, and this can be tough for patients with encephalopathy. And then, of course, also we required an expensive analytical equipment and highly trained staff, but also is a timing problem because, as we know, the drug level fluctuate during the day, and a single hospital blood draw only provide a snapshot, so it doesn't reflect the patient's real-world daily exposure. And this is why TDM in the future can be shifted to decentralized at-home sampling to monitoring patient in the exact moment of the seizure. So shifting to at-home monitoring is possible, can be possible, but with the use of capillary microsampling devices. These are that technique that are based on a finger prick, so a little prick that can collect a small volume of capillary blood. And using a finger prick, a patient can collect a drop of capillary blood self collection. And of course, we have different advantages. For instance, we can reduce the sample volume, is minimally invasive. The procedure is pretty easy, so they don't need training. They can even do in uncooperative patient. There's also the stability of the drug at room temperature, and so it can be possible to send by regular mail to the laboratory. And this drastically can cut down the patient travel cost and act with an excellent support tool for telemedicine. Nowadays on the market, there are different devices. Here I reported the main, the most used. As you can see, there's EMA-Pen, VAMS, QDBS, and then there are the most new, there are MAXIS, and the last one that is it's pretty new, and this is the touch one. So for all the research that I did, that we perform, we focus our attention on VAMS technology, that is volumetric absorptive microsampling. And as you can see, these devices consist in a plastic handle with a specialized polymeric tip. And the key innovation here is the volumetric absorption, because unlike a filter paper, here there's a blood drop sites that could not bias the result in theory, because the tip should absorb an exact and precise micro volume, and they can either absorb 10 or 20 microliters. And for all the validation and the research that I will show you later, we use the 10 microliters format. But here there's also another device that we also use, that is quantitative dried blood spot, so-called QDBS. And this is made from a little tip, a little disc, where is a pre-cut, so we can remove and use for the analysis, and should be easy for the patient because it collects an exact volume that is again 10 macroliters. So how we can read the result? So first of all, we have to use the extraction method validation. So we have to perform an extraction validation from a specific drug. Second, we have to compare the blood to the plasma and also consider the hematocrit effect. And third, we have to convert the capillary dried spot into the standard plasma concentration value. So to foresee if they are compared. So this brings us to the third part of our presentation, that is focusing on the active research project that was designed and validated for the reliability and real-life feasibility in these microsampling devices. So I will present the main project I work on. So we first of all work on microsampling for telemedicine. Then we work on at-home sampling. And now we are working on a POP-PK, so pharmacokinetics population. So the first study that I would like to discuss is the microsampling in telemedicine. So the primary aim was to evaluate the integration of the telemedicine into epilepsy care. Specifically tracking the transition from traditional office-based visit to video consultation in terms of seizure control. But also we wanted to implement VAMS as an alternative method for these patients and compare nurse collection versus self-sample collection to the traditional venipuncture. So we did a double cross-validation. Then we integrated the VAMS sampling result into direct the patient remote video consultation in order to improve the treatment adherence and to reduce the number of the hospital visits. So this, as I said, was a cross-validation study that was designed to compare three sampling methods: the traditional venipuncture, the nurse collection VAMS, and the self-collected VAMS, that were performed by the patients in the ambulatory setting or by the caregiver. All samples were analyzed through HPLC-MS method, and a statistical agreement was evaluated using two methods. That was a blunt Allman analysis that can evaluate the agreement and the systematic bias between the two measurement methods and verify if their difference fall within a clinically acceptable limits. And then a passive Bablock regression, that is a linear, robust linear regression that can measure the correlation and proportionally between the two methods. So to better understand if the data distribution and also the potential outlier. So this was the results of this project. We enroll adult patients on chronic antiseizure medication therapy who were capable of performing the capillary sampling independently or with an available caregiver. We evaluated more than 300 patients with a mean age of 44 years old and with a slightly percentual predominance of female. We're in 50-50 monotherapy or polytherapy. And we had to exclude 60% of the samples due for a primary underfilling or overfilling the microsampling. And looking at the single drug, lamotrigine was the most frequent monitoring with more than 100 measurement. And this actually reflects our clinical data. So in this slide, I showed analytical validation and the agreement of lamotrigine concentration. So in the upper side, we can see the venipuncture, so the plasma, versus nurse collection VAMS. And the blunt Allman plot show a mean bias close to 0, meaning that the bias was practically non systematic, over or under estimation. And the passing Bablock regression display show an excellent linear correlation with the regression line, tracking the very close and ideal identify line. So the crucial part of our study was the lower section, because we also compared the venipuncture versus the patient's self-sampling. So even when the patients perform the finger prick entire by themselves, the results remain robust. And this was the mean bias, so the bias was close to zero, and the regression line proved that the self-collection was accurate, just as the same collected from nurse. But if for lamotrigine we found a good correlation, this is not possible for other drugs, for instance, perampanel, topiramate, and valproic acid, and zonisamide, where, as you can see in this slide that show again Blunt-Hellman analysis and passing Bablock regression, we saw that the bias was suboptimal, and meaning that the VAMS utility is limited for this kind of drug. So we could demonstrate the reliability and feasibility for 7 drugs on 13. And that include lamotrigine, as I show you, but also carbamazepine, lamotrigine, and phenobarbital. Here is another slide where I want to show the nurse VAMS versus self-sampling VAMS. So again, we did the cross-validation from nurse and self-sampling, and this sub-efficacy is likely due to the specific drug properties. So these can be from high plasma protein binding, so significative hematocrit dependent distribution, or the need to more tailored extraction protocol from the lab. So these actually tell us that the VAMS require a really compound-specific optimization. So even if there are the good correlation between VAMS and self-sampling, the same drug that we couldn't extract properly from nurse, it reflects also from self-sampling. And this is crucially for telemedicine, because we actually found a strong correlation between sample collected from nurse and self-sampling that confirm the user-friendly nature of the microsampling. But we also known that we need to better understand the other drugs, why they don't fix. So additionally, the drug stability test show that we found a compromised for all the drugs that was a 7-day window before the analysis to the lab. This brings us to the second part of our project, that was the at-home microsampling, where we wanted to perform directly at-home patients. So we evaluated the analytical reliability and at-home VAMS versus the ambulatory plasma. And we also assessed the feasibility through a specific survey. So here is the method. We first collect the standard venipuncture, so the plasma in the ambulatory. We did the VAMS in the ambulatory again, and the self-collection VAMS was this time was just performed from the patients, so not from the nurse. And then we ask the patient, we give them a kit, and they did the exact same collection the day after at home, and send the kit, so they send them microsampling to the lab the day after. And Still, we analyzed the devices through HPLC-MS method, and the same statistical analysis were performed, so the Blunt-Ullmann and the passive Bablock regression. Here, regarding the feasibility, I want to show you that we use this survey where we ask regarding the pain that they could perceive, and how easy was the method for them, and how good was the shipment to the lab via regular mail. So what we saw in this result was the clinically application and the demographic of the at-home cohort. So we include 103 adult patients still on chronic therapy on antiseizure medication. The population actually reflect the previous study, so the real-world age range, so from 19 to 87 years old. Majority this time were women on monotherapy. And on the right, the pie chart displayed that our 4 monitored drug with lamotrigine that represent again the most frequently used prescribed drug. I also show you an MRM, that is a chromatogram on the right, that it provides an example of our analytical separation of this compound to LC-MS. These are the results from the VAMS at home versus VAMS ambulatory. Also this time I show you Blunt-Ullmann analysis and passive Bablock regression for lamotrigine. And as you can see on the left side, the Blunt-Ullmann analysis again demonstrate a good agreement between the two settings, and the mean systematic deviation was minimal, and the majority of the patients fall within the limit of agreement with only one outlier, confirming actually that the shipment from the ambulatory to the at-home environment does not introduce any significant analytical bias. And on the right, I show you that passive Bablock regression that still highlight the linear correlation. So on the summary box below, this direct comparison proves that VAMS devices are highly reliable for the at-home use, and this actually open a possibility for TDM at home, so decentralized. Then we speak now about feasibility. Then was performed using the survey that I showed you before, and more than 80% of the patients, of the samples, reach the laboratory in 7 days. That, as I said, was the therapeutic, was the time window that we decided before the analysis, so for VAMS stability. And over 80% were actually proper collect. But I want to say that actually every patient receive a dedicated face-to-face training section and how to use the VAMS in a correct way. And we actually also call them on the phone on the scheduled day of the sampling. We remind them the procedure and help them with the delivery. And 90% of the patients also found that the procedure was easy, minimally invasive, painful. So we actually could confirm that home sampling is feasible in a real life setting. And from a logistical standpoint, the process provides highly feasible. So over 80% of the samples arrive to the lab in the time that we decide. And the satisfaction was pretty high. 90% said that was easy to learn, little pain. And so we think that this can be vital for the long-term treatment adherence in telemedicine protocol. Now we want also to show you another part of the project that we did with the second device that I present you before, that is the QDBS. So in this case, we also did a cross-validation through QDBS and venipuncture, so plasma. So in the ambulatory, we did a standard venipuncture to get the plasma. The patients performed the QDBS and the VAMS. We analyzed all with the same analytical method and with the same statistical method. Yeah. And what we wanted to see this time was first of all to validate the method for the QDBS, because it wasn't performed in literature. So we wanted to validate the method for all the drugs, for all these 4 drugs that I showed you before. So here I just present you the analytical part, and this is the clinical validation. So we use clinical data. So we collect the drugs from the blood from 20 patients for do the clinical validation. And then we perform the reliability of the QDBS and VAMS. And also here we can show that the 2 methods, the 2 microsampling can be compared with good Blunt-Talmant and Passing-Bablock. And now we pass to the third part of the project, that is the POP-PK. So what is a POP-PK? Population pharmacokinetics is a model that we decided to perform for lamotrigine, because as you see until now was the drug that we could better use with the microsampling. And we use a robust and clinical data set from TDM that was performed in Swiss cohort at the CHUV, so the University Hospital in Lausanne. And POP-PK is actually a mathematical and statistical modeling approach where we can study the drug concentration in a target patient population. And with this model, we can calculate the average of pharmacokinetics value while strictly quantify the variability between the individuals. So we can actually better understand if the model is robust, and with the final aim to use this model with VAMS approach. This actually why we choose to use POP-PK. This is actually because the traditional pharmacokinetics require multiple frequent blood draw from the same patient, but this is actually impossible in at-home setting. So POP-PK can help us to design a model with the spare data and build a predict model using just 1 or 2 VAMS samples for patients. So use POP-PK, we can identify how specific covariants like patient's age, weight, or concomitant therapy can affect clearance and distribution volume. And as I said, the final aim was to perform this model from the precision dosing. So actually to allow clinicians to use the VAMS concentration and building on the model from TDM data. So what we did here was to create a reference model from CHUV dataset, do an external validation with the TDM data in Italian dataset with plasma, and then once we had this and tried the various co-medication, validate as well the model onto VAMS microsampling data and see how to use this model for VAMS. So what we did, first of all, we had to clean the data, because who works in TDM knows that data are also really not clean, because it takes in routine. So we needed to perform a real good cleaning before to use this data for building a model. And then we use this data for build the reference model. And actually, there was a pretty large cohort in Swiss, because they do TDM in epilepsy for routine. So we had more than 350 unique subjects, so more than 600 patients samples concentration. And this was really useful for perform the method. The population in this case was with a mean age of 43 years old and with a body weight of 62 kilo. This, I just tell you this because it's really important when we go to see the Italian data set. And so with this, we could actually start the building of the model. These are the preliminary results, because I'm still working on this model. So what we found was we built a one compartment model with the oral administration that was characterized by a first order absorption and linear elimination. And to explain the pharmacokinetics variability along patients, we test the several covariants, specifically age, sex, body weight, and to the statistical stepwise process that is forward selection and backward elimination. The body weight was emerged like the only significant covariant that impact on both clearance and volume of distribution. And the final model that is specification, this is in the bottom, is where the weight was integrate into the model with our median population weight of 62 kilos. So for this, we did this analysis. So the population clearance we found at 1.89 liters per hour. The volume of distribution that we found 136 liters. And we saw that both were depending from weight. And this mathematical extraction actually allowed us to tailor and predict drug exposure based on patients' body weight. So looking at the right, there is the visual prediction check that highlight the high predictive performance of the model. So the solid line represents the experimental percentile of our data. And as you can see, they lie within the blue and pink shade area that represent our model 95% confidence of interval. And this graphical design can confirm that our model is ready for the external validation that we did here with the Italian data set. So the model was considered power. We performed the external one with the Italian cohort of 37 patients and 120 plasma sample. And interesting was that the Italian cohort was younger and the body weight was a little less, was like the median was around 60 kilos. But when we went to the validation, the true one, the external validation, we could see a solid and robust predictive performance, because the mean prediction error that is so-called bias was 11%. So it indicates a modest clinical acceptable tendency to slightly overestimate the real plasma concentration. And the overall precision under real-world clinician condition was reflected by a root mean squared error that was 39%. That compared the final model can be considered stable. That's really important because we're a completely different hospital data set. And here I show you the goodness of fit, that is the observation data versus the individual prediction data. So, which are the next step? Actually, we are not we didn't finish yet this project because we wanted to test the K covariant, so the co-medication drug, because of course we wait that we expect that the enzyme inducer like carbamazepine or phenytonin, and we saw that some of the patients are also in polytherapy, go to increase the lamotrigine clearance, and the opposite way around is regarding the enzyme inhibitors. And also we want to see the pregnancy, that is another covariant that we have to consider when treat the patients, because also in pregnancy we wait that the clearance of lamotrigine can increase. And last but not least, of course, we wanted to identify an alpha correlation coefficient that we wanted to use when we have the VAMS concentration. And so first of all, we want to externally validate the model using the VAMS database from Italy, and then establish the direct mathematical transition from the capillary blood concentration back to plasma values. So before we go to the conclusion and hoping for the future, of course, we have to talk about the study limitation, because of course, what we have found out was that, as I show you, some drugs, for instance, valproic acid, is suboptimal extraction, show a suboptimal extraction recovery in dried matrices, and then is actually cannot be used for this analysis, or at least we have to better study the use of these drugs for the microsampling. And then of course there's a different kind of matrices, because of course the microsamplings mitigate the volume hematocrit effect, but the potential inter-individual variation in patient hematocrit and the blood to plasma ratio can also still introduce the analytical bias. So we have to consider how to convert the whole blood into the plasma value. And then there's also the small sides of the device comparison, because for some of the drugs that I didn't show you because I showed just the lamotrigine, but for other drugs we didn't have so many patients, so we had to exclude the drug for this reason. And also performing the finger prick at home introduce a potential real-world variability, for instance, insufficient sample volume, as we can see in the first project, or incorrect driving times or storage. Regarding the retrospective design, first our reference model was built on a retrospective real-world dataset from SHUV. So this means that the sampling times were from a routine clinical practice that is not, of course, in a strict protocol. And so naturally, we can introduce some noisy and heterogeneity into pharmacokinetics data. Second, the Swiss cohort was robust, but the external validation cohort, specifically in the VAMS dataset, is smaller, and this can limit our ability to run deeper the analysis in this stage. And third, as I mentioned earlier, the high impact of covariant like pregnancy and drug-drugs interaction is still ongoing. So then we have to actually see the study limitation of the use of microsampling. This regarding the realistical condition. So who supervises the sampling at home? We cannot see and we cannot call the patient for every step, because it's not possible in real world, of course. can be possible to perform in low or middle out country, where we can see that no one, of course, do TDM, but maybe we can use a device in this country and analyze in Europe, but we have to understand how to transport the microsampling. And then we have to consider the patient motivation to perform the device. For instance, do it at home and then send it to the lab. So we have also to see if they are enough motivated for do it. And then, of course, the laboratory needing, because there is no kit available for microsampling devices yet. And this, of course, slow down the laboratory lab routine. And also at the end is just about money, of course, because there are not so much funding for research. The device costs a lot. So also when we had the device, the laboratory staff cost, and we have to understand who do this and study for do this. So of course funding is it's a really study limitation for all. And then of course understand who pay and who saves regarding the health systems, for instance. So of course the microsampling can be used for remote area, or can improve the compliance, or can reduce the side effect, or improve clinical outcome, because we can potentially use in the breakthrough seizure and reduce the emergency hospitalization. But all of these need to be better studied. So just a bit of hope before leaving. What we did until now was a part of a big project, and of course we found some things that was good, like the excellent linearity and precision for some drugs, but of course the success of at-home patient also can be considered a good result, and the integration of capillary to plasma conversion in POPPK model can also improve the precise data-driven therapeutical dose adjustment. But the next step are a lot. For instance, we can use the microsamplings for a finger pink. So the application in this special population, for instance, pregnancy, when women in epilepsy have to do the TDM really often, or use the device in different pathologies. Here I just report the lyso-Gb3 VAMS, or also for levodopa in Parkinson disease. But obviously also we want to, as a future need, we want to move more into real life data and clinical trial that are required to demonstrate the long-term clinically and economics benefits of this practice. So it's all to be continued. And with this, I want to thank you for the attention, of course. Thanks the Centro Epilessia of the Istituto delle Scienze Neurologiche di Bologna, where I work, and the University of Lausanne and the hospital in Lausanne, where I'm doing the POPPK right now. So thank you so much.
Thank you, Chiara. That was very, very interesting. We've got a few questions coming through. I think a bunch of the questions have been answered as we went through. So I'm going to concentrate on maybe some of the ones that you didn't cover quite so much. Question here from Rona. How did you overcome hematocrit effect or evaluate it?
Yes, we did with all this data, and we found a mean hematocrit effect. So we used that data for evaluated. So yes, we did. We found that it doesn't really affect the data, but it's something that still we have to do. So even if this microsampling should avoid this problem, we also do. Yep.
Okay, thank you, Chiara. Question from Jason. How is the adequacy of filling determined?
Sorry, didn't understand the question.
How is the adequacy of filling determined? So I assume on the VAMS microtips, how do you determine whether it's been underfilled or overfilled or correctly filled?
So we decided to give them a quality score. So double blind, two persons, so me and my colleague, see the VAMS and give a score from 0 to 5. So 0 if it was under filling, and 3, 4, 5, like 4, 5 was if it was a good filling. So we did, and then if wasn't the same number we talk about, and we decide what number give to that microsampling. And then we actually, the second part, so the at-home one, we did the first correlation with all the VAMS, so from 0 to 5, but we saw that there wasn't a correlation, of course, because the 0, 1, 2, and 3 are kind of empty. So then we decided to keep just 4 and 5 as a quality score. I think there was a graph that showed the percentile of the quality score. So a lot of them were on 5, even at home, even in the ambulatory, but still there was a small percentile underscore.
Thank you, Chiara. Question from Yan Chun. How were the patients recruited for this study?
The patients were recruited, so they were adults because we work in an adults ambulatory. They had to be able to perform the VAMS independently. So in the first study, since there was also the telemedicine study involved, we decided also to involve the caregiver. So we asked the caregiver if they couldn't do it, for instance, the encephalopathy, the caregiver also did it. But in the second part of the study, where they had also to do it at home, we just we just enrolled the patient that were able to do it by themselves. So this was the criteria, actually. And then since, of course, they have to do the venipuncture and the microsampling before taking the drugs. So we actually choose the patients who were early in the morning.
Okay. Thank you, Chiara. I think we have time for one last question from Jason. Any study done on the effect of temperature on the stability of the analytes?
Yes, we did a lot of study for the temperature. We actually saw that for some drugs there was highly stability in minus 20 degrees. But what we wanted was a real world data, because of course you cannot ask the patient to keep the microsampling in the freezer or to send them in a freezer bag. So that's why actually we wanted to find a compromise for all the drug at room temperature, because for us was a data that was more usable for real world. So for some drugs, yes, there was a good stability, also even a high temperature, for instance, but wasn't for all the drugs. So for find a compromise, we decided 7 days before the analysis. That this is not a lot of time, because the patient need to send the sampling, and you need to analyze in a small time, but that was the time that we found most stable for all the drugs.
Okay, thank you, Chiara. Just thank you, Karina, for a quick clarification about noting about filling of samples. She said you can visualize if the VAMS tip is fully filled by verifying that there are no remaining white sections. If underfilled,
Yeah,
it can be
exactly.
applied again to a new blood drop to finish filling. So I just wanted to put that in as well, Chiara, to add to what you'd said. So, no, thank you to everyone for your questions. I'm just going to throw up some slides to finish with. These webinars are brought to you by the Patient-Centric Sampling Interest Group. We're a not-for-profit group, and we're here to educate people about patient-centric sampling and work out what the challenges are to more widespread adoption of these technologies for patient benefit and figure out how we overcome these challenges. If you want to learn more, the QR code on the left-hand side takes you to our website. The QR code on the right-hand side takes you to our LinkedIn group, where we post information, share conversations, ask questions, etc. So please, if you're interested, ask to join that group. Our next webinar is coming up in July the 13th, with Tim Woolley, and it should be a very interesting one. It's an introduction to the UK recommendations for the validation and adoption of capillary blood testing within the routine clinical laboratory. And then we won't be having any webinar during August because a lot of people are on holiday, and we'll return with webinars in September. So with that, I'd just like to thank all of our partners and sponsors again for providing the funding for PCSIG so we can bring you these webinars and the content on our website. I'd very much like to thank Chiara for her wonderful presentation today, and I'd like to thank the audience for your great questions and your attention. And I'd like to thank Phil and his colleagues at Bacaris behind the scenes for making all the technology work. And that will finish. Thank you, everybody.